Welcome back everyone, this is Preeti from Hassan Institute of Medical Science. In this video, I'll be discussing about further the functions of sympathetic and parasympathetic nervous system and the cholinergic transmission, how the choline is synthesized and transmitted, and the types of cholinesterase and cholinergic receptors. In this video, I'll be covering all these things. In my previous video, I covered about the difference between somatic and autonomic nervous system and central nervous system overview. If you want that video, you can find in my description. Now I'll be teaching about the sympathetic and parasympathetic, how to remember. Like, you need not to mug up anything, you just need to imagine a situation so that you will remember all the functions of these two. First, you now take parasympathetic nervous system. Parasympathetic nervous system is when you are not doing anything, you are just simply see, like this baby, you are just drinking a milk and then sleeping, so relaxing, and you are drinking this thing, so it helps in digestion. Parasympathetic helps in digestion and for relax when you are not doing anything. So it increases GI motility and secretion. This is for digestion, it helps in digestion, so it increases GI motility and increases secretion, which further helps in digestion. And sphincters are little relaxed, so you defecate when you are very calm. Defecation or urination, all this comes, urination or defecation, all when you are calm you will do defecation. Even though if you are in a emergency situation, even though if you want to do defecate or urinate, you will control it during that time. So in sympathetic nervous system, this sphincter tone is more increased, that is sphincters are contracted, whereas in parasympathetic parasympathetic stimulation, the sphincters are relaxed, so you will defecate or urinate in that in parasympathetic under the parasympathetic stimulation. So you learnt about a GIT. What is the action of parasympathetic nervous system on GIT? That is, increases GI motility, increases GI secretion, and sphincters are relaxed, so you defecate or urinate. This is a parasympathetic stimulus on GI muscles, that is smooth muscles of gastrointestinal tract. On this parasympathetic situation, that means you are not in an emergency situation, so you need not want that much oxygen. Like when you see snake or something, you should run, you need energy for that. Energy means you need energy, means you need ATP should be produced only during oxidation process, oxidative phosphorylation, ATP is utilized and then energy is produced, that is ATP is produced. Oxidative means using with the help of oxygen. Phosphorylation is production of ATP, so for that and all oxygen is required. But in parasympathetic, you are so just relax, you need not to require that much energy. So bronchial constriction will be there, so entry of oxygen into the lungs will be limited. The bronchial constriction will be there as the energy demand is less. And when you see like this, see, when you are when you are scared for something, pupil dilation will be occur, that is mydriasis will occur. But when you are calm like baby, the pupil will be constricted. So meiosis will be occur in parasympathetic stimulation on the pupil. Meiosis will be occurred because of the constrictor pupil. It acts on the constrictor pupil. The eye will be having this circular concentric muscles are constrictor pupillae, whereas these radiating muscles which dilate the pupil are called the sphincter pupillae or radial pupillae. The circular muscles are sphincter pupillae, whereas the radial muscles are dilated pupillae. These two muscles will be there based on these stimulus. There will be meiosis and mydriosis in parasympathetic situation. Parasympathetic stimulus, when there is see, the baby is calm, so the pupil is constricted whereas this is they have downloaded from the cartoon. This cartoon images where they are scared for something, so the pupil are dilated. This condition is called mydriasis, whereas for pupil constriction is called meiosis. And you learnt about the function and the stimulation of parasympathetic on bronchial, on GIT and on eye. Now we will discuss about on the heart, how the parasympathetic stimulation stimulus results in the heart. Like heart means parasympathetic means you are not, like you are calm, so heart rate will be less. Heart rate is less. Force of contraction of heart muscles, that is atrial contraction and ventricular contraction, will also be less. I think by listening to this, you already remembered everything, because I told like a story, and then you imagined a situation where you come across all this, so you remembered everything. And GIT, GIT secretion will be increased, and GI secretion will be increased and motility is increased, and sphincters will be relaxed, so that defecation and urination will be occurred. This is on GIT. And then bronchial muscles, for bronchial constriction will be there, because it doesn't require much energy, much oxygen, so bronchial constriction will be there. And on I, meiosis will be there. We are calm, so pupil is not dilated. And then heart, the further pumping of heart is not required, not much energy is to be pumped, so heart rate is decreased, force of contraction is also decreased. And this is all about the parasympathetic nervous system. Now imagine a situation where you are in an emergency situation, like you have to fight against that situation, or you have to escape from that situation. You have to escape means you have to run, no? Run, that is flight, or fight, when you have to face it and fight. That situation, when you see think that you saw a snake, you have to fight against that, like you have to kill that snake, or else you have to run from there. So you need energy. For if you run, oxygen, like all muscles started working, the requirement of that muscles, oxygen demand is also increased, so oxygen entered the trachea. This is a tracheal. This trachea this is the trachea contains bronchial dilation. After trachea there will be bronchioles. These bronchioles undergo dilation, so oxygen is more entered. This is you learnt about this. And mydriasis, as I shown in this image, they undergo, when you see you are frightened, your pupil will be dilated. This is mydriasis. You got these 2 points. And increase in the supply, like energy demand is increased, so oxygen supply is also increased, so heart rate also increases, force of contraction is also increased. And GI motility, during when you are emergency situation, you need not to defecate or urinate, that sphincters are constricted, or sphincter tone is increased. GI motility and secretion is mainly under the parasympathetic control, so this motility and secretion is also decreased. This decreases. This is all you learnt about the sympathetic and parasympathetic. I hope you properly got all these points, so you need not to mug up anything. So just imagine a situation where the baby is rest and drinking milk. In this condition, where you saw a snake and you need to fight or flight. This is all about the sympathetic and parasympathetic nervous system. Now we will see the cholinergic transmission. How choline is synthesized, stored, and then released into the neuromuscular junction. And now this is a nerve ending. You think this is a neuromuscular junction, or better say synapse between the two neurons. See, this is also one neuron, and this is also neuron. Between the two neurons, that is, we take as a synapse. This is a presynaptic membrane. This is a postsynaptic membrane. Postsynaptic membrane has receptors for acetylcholine. First, we will see how the choline is entered, like how acetylcholine is produced. Now we will see how acetylcholine is produced, stored, and then secreted into the synapse. First, mitochondria have acetylcholine. As you know, mitochondria has acetylcholine. For the TCA also, this is a main substrate, so acetylcholine will be there from the mitochondria. This acetylcholine comes from the mitochondria. This choline enters now presynaptic membrane through the this enters. This is entered by carrier-mediated transport. From carrier-mediated transport, the choline is taken into the nerve fiber. This acetylcholine, which is from the mitochondria, and this choline will combine to form acetylcholine. Acetyl-CoA and choline combine to form acetylcholine. Acetylcholine by the enzyme choline acetyl transferase. Choline acetyl transferase. It is converts into transferase means it converts into acetylcholine. Choline acetyl transferase, which converts into acetylcholine. From this acetylcholine is picked up into the vesicles, and then it is stored, and then during the release it comes and attaches to the active site of this presynaptic neuron, and then this comes and diffuses with this presynaptic membrane, and then it releases acetylcholine into the synapse. Now, the fate of acetylcholine, if you now I said about the synthesis, that is choline and acetyl-CoA will combine to form in presence of acetylcholine transferase to form acetylcholine. This is a synthesis. Storage, this is stored in the neuromuscular vesicles in the presynaptic membrane, and then they diffuses with the presynaptic membrane, and then they are released out into the synapse. Now we will see about the fate of this acetylcholine. How what are the fate of this? What will be, how it will be changed? There will be three conditions. It may be picked up by the this receptors. Receptors for acetylcholine will be muscarinic and nicotinic receptors. Any one of these will be picked by this. Acetylcholine is picked up by this receptor. This is one fate, and other will be, it will be taken back into the presynaptic membrane, that is called autoreception. This is the same neuron will takes this thing, so this is a autoreceptors. And other fate will be, it will be hydrolyzed acetylcholine, no, again it will be hydrolyzed to acetate and choline by the enzyme acetylcholine esterase. Acetylcholine esterase. AS means enzyme. That AS, whenever we see AS, these are enzymes. The full form of ACH I have written everything short, so I have written separately. ACH is acetylcholine. Choline esterase, choline acetyl transferase. This which helps in converting acetylcholine and plus choline into acetylcholine plus choline into acetylcholine. And this choline acetylcholine esterase which break down acetylcholine into choline and acetate. This is all. These are cholinergic receptors. The fate you got to know that it is taken up by the receptors or it is taken back by the same neuron, that is autoreception or hydrolyzed into choline and acetate. This is the fate of cholinergic transmission. And now we will see about the choline esterase. Choline esterase is also known as acetylcholine esterase. Shortly we can say choline esterase or acetylcholine esterase. As we have discussed in the previous page only, this choline esterase hydrolyzes acetylcholine into choline and acetate. See, choline esterase hydrolyzes acetylcholine into acetyl and choline, acetyl or choline or acetate or choline. There are two types of choline esterase: true choline esterase and pseudo choline esterase. True choline esterase are also known as acetylcholine esterase, pseudo choline esterase or butyryl choline esterase. This is you need to know the difference between those two. The site will be different, and which will it will hydrolyze which one, that will be different. The site where are present where true choline esterase are present is the site where this cholinergic neurons true choline esterase are present, and the site where pseudo choline esterase are present. You can remember through this mnemonic, like C ring. This C is a one thing. Only I is not there. In C ring, only I is not there. C, R, N and G. For C, cholinergic neuron. In all cholinergic neuron, this true choline esterase or acetylcholine esterase is present in cholinergic neuron, RBC, this is a ring RING, ganglion and neuromuscular junction. This is cholinergic neuron, RBC, neuromuscular junction and this ganglion. C ring, you can remember the mnemonic for the site of where you locate the true choline esterase. And for the pseudo choline esterase, you can remember as plug, PLUG. P for plasma, L for liver and G for glial cells. Glial cells are the other types of in nervous system we find other types of cells other than neurons. These are glial cells which are supportive cells for the neurons. This you can remember through plug, PLUG, plug. And the substances which are hydrolyzed by the true choline esterase will be acetylcholine obviously and methacholine. Both are hydrolyzed by true choline esterase. Whereas pseudo choline esterase or butyryl choline esterase, it can hydrolyze only acetylcholine, not methacholine. Acetylcholine also it is hydrolyzed only very slowly. It hydrolyzes acetylcholine very slowly, whereas it cannot hydrolyze methacholine. This is all about the types of choline esterase. And then now we will discuss about the cholinergic receptors. What are the receptors present in the choline? For choline, acetylcholine is in C. Now this acetylcholine has entered into the postsynaptic membrane. This should be received by some receptors. No, what are the receptors present? Choline which is released from the presynaptic neuron should be received by some receptors on the postsynaptic membrane. This receptor, what are the receptors we will see, that is muscarinic and nicotinic, and further the subtypes in muscarinic and nicotinic we will see in this. Cholinergic receptors, there are two types of cholinergic receptors, that is muscarinic receptors and nicotinic receptors. All muscarinic receptors are G protein coupled receptors, whereas these nicotinic receptors are ion channels. They directly open the ion channels, whereas these produce secondary messengers. There are two types, like this you consider this as a cell and this is a nucleus. For some the receptors are present here only, and so it acetylcholine comes and binds here and further produces a secondary messengers like calcium, IP3, DHE. All these are secondary messengers. This can be produced like if the receptors are on the outside membrane, or they can be directly ion channel. It opens ion channel, so ion entry will be there will be depolarization, that is calcium or anything, potassium, anything. There will be depolarization. There are two types of this thing, whereas G protein coupled receptors, that is muscarinic receptors, produce secondary messengers like IP3 and DAG. These are the secondary messengers, so they can be produced, whereas nicotine directly open the ion channels. We'll see in detail in my further videos. That is, now we will only discuss about the subtypes of these types and subtypes of these receptors. Muscarinic, again we have 5 subtypes, that is M1, M2, M3, M4 and M5. Out of these M1, M2 and M3 are functionally important, whereas M4 and M5 we don't discuss much about this, but the location of this will be in CNS. On CNS, in the CNS we'll see M4 and M5 receptors, whereas M1, M2 and M3 we'll see where it is. You need to remember this so that it will be helpful in my further videos. That is, M2 are present only in the heart will see M2. Only in heart we'll see M2 receptors, whereas M1 receptors you remember like CNS, the brain, and then in the central nervous system, brain, and then in the autonomic nervous system, the ganglion in the autonomic nervous system, M1 mainly in the gastric gland, GIT. Whereas in all except GIT, all smooth muscles have this M3 receptors, all smooth muscles except GIT, and then exocrine glands and endothelial cells, means blood vessels have endothelial cells. The lining epithelium of blood vessels are endothelial cells. Endothelial cells further secrete nitric oxide. These receptors are present in the blood vessels. So M2 only present in the heart, M1 CNS, autonomic ganglion and gastric glands, whereas M3 all the smooth muscles except GIT, and then exocrine glands and endothelial cells. M4 and M5 CNS, whereas nicotinic receptors further they are subdivided into 2 groups, that is NN and NM. NM, as the name says, neuromuscular junction, near neuromuscular junction NM receptors will be there, whereas NN in autonomic ganglion and CNS is same as here. In autonomic ganglion, this is as a ganglions. Here both N receptors are there and M1 receptor also there in CNS and autonomic ganglion. And NN, this particular will be, this is a kidney. On the top of kidney will be having adrenal gland. On the adrenal, this is a adrenal cortex and this is a adrenal medulla. Here adrenal medulla we have NN receptors. These are the 3 sites where you will find NN receptors. They directly open the ion channel further action. If you find this video helpful and you can easily understand what I am saying, please stay tuned to my videos, and please encourage me for your encouragement will be for me motivation to do further more videos. Please do like, share and subscribe to my channel, and thank you for watching till now.